AI has been all the rage, but how useful is it really? To get a sense for that, let's rewind about a decade into the past when dozens of undergraduate students in our laboratory would work together day after day, putting in hundreds to thousands of hours, counting images of the small white eggs that female fruit flies lay, in an effort to quantify their fecundity. These hours were spent just to make the large-scale experiments using this metric viable, and it was so time-consuming that to this day, there's uncounted data that still needs to be analyzed if only we had the manpower to do so. In this project, our goal was to intelligently use AI to replace this menial work, and ultimately, we made fecundity assays more productive without sacrificing the accuracy of our data. Why fecundity? Fecundity is a crucial metric in evolutionary biology because it's used to estimate the biological fitness of a population in certain environmental conditions. These conditions can be molded to test various treatments, such as drugs or different diets, so fecundity matters to any lab hoping to examine large-scale population change and adaptation to these conditions. And as you can imagine, the previous method was too labor and time-intensive to be conducive to large-scale data being analyzed efficiently. To fix this, we proposed a novel machine learning model built specifically to count eggs in an image. And initially, we scanned the internet for ideas, but we're disappointed to find that there were no up-to-date tools that worked for the task. So we realized we would have to make our own tool, and in fall quarter, we joined forces with a computer science student in our lab, expanding this into an interdisciplinary effort. Initially, we knew we wanted to take advantage of the burgeoning advancements in machine learning and computer vision. After refining our search, we decided to develop a convolutional neural network, which is usually used for tasks like facial recognition, but we use it to detect and count eggs in large sets of images. By telling the computer how different groups of eggs look, we created an AI that automatically and accurately estimates female Drosophila egg counts. With this done, we drastically reduced the time and labor needed for gathering fecundity data, while minimizing the potential for human error. This exciting new technology has broad applications for fecundity assays, both in our lab and any other biology lab that works with Drosophila as a model organism. This tool will make fecundity assays vastly more accessible to perform on large sample sizes, and thereby allow labs to do larger scale experiments with more statistical power by freeing up the time and resources through automation. Thank you very much for your time, and I'm excited to present our work in depth at the UROP Symposium. We'll see you there.